actually advocating live attenuated vaccine. However, there are dead viruses, or the main one is gene recombinant, which means that they cut the gene that produces the spike protein, inserted in the nucleus or in the gene product. Actually, the virus has got a nucleus in the genomic structure of a virus like adenovirus, human or chimpanzee, or an animal virus like the vesicular virus. And these viruses used as a vehicle to produce the spike protein to stimulate the immune system without doing any harm. Now, other vaccines like protein combination or viral particle, which is genetically measured by having the viral code and the structure, and because the whole point is to put into the body something that have the similar antigenic structure of the virus to stimulate the immune system. Now, we got a new technology, which is really brilliant. We call it messenger RNA. Next slide, please. Next slide. Now, I think I have already talked about the traditional vaccine, which are mainly either protein or virus, which contain part of the genomic structure that produce the antigen. And then on the left-hand side, you can see the messenger RNA vaccine. Next, please. Messenger RNA vaccine is using nanotechnology, which is really a clever way of doing things. In fact, this nanotechnology has been used for so many years now to treat myeloma. And in fact, some of the antibiotics has been used, like liposomal amphotericin or amikacin, used in cystic fibrosis patients and other patients. The whole point about messenger RNA is they call it genomic editing. As you know, the function of the cell depends on the genes. The genes are inside the nucleus. What express or make these genes expressible is by sending the messenger RNA, telling the ribosome to do enzyme, hormones, proteins, anything. And some people are worried, or some people actually falsely calm by saying, oh, this messenger, don't worry, it doesn't come inside the nucleus. In fact, they should know that the cloned traditional vaccine, they are actually, the DNA has been inside the genomic structure. So this one is outside. But this is a very important thing, and it's important thing from the safety point of view. Next, please. Now, this is a good illustration about how messenger RNA work. And when the synthetic viral messenger goes to the ribosome and it translates, tell the ribosome, actually, you must do the S protein. And the S protein hence appear on the cells. So this is, as I said, a clever way, and this is one of the easiest vaccine, and it can be given, in a very much convenient way. Next, please. Now, going back to the experiments, as you know, those who are doing research, everything will succeed in a test tube. Less so in animal studies, and very much less so with human studies. Hence, the phase 1, phase 2, phase 3 clinical trial. And the reason for this clinical trial, because to fulfill the effectiveness as well as the safety, and usually, as you know, case control studies, because each study has its first endpoint, which is the laboratory finding or the biomarkers. And the second one is how the result affects the patient, whether it's life expectancy or quality of life, rest of it. Next, please. Now, whom we get the vaccine to? This is based on risk assessment with the help of the statistician and modeled by the epidemiologists. Because statistics tell us a fact about distribution of the disease. Epidemiologists tell us from their previous experience with other diseases how you can model your effective way of vaccinating the people or preventing the disease. Next, please. How confident one can be about a vaccine. We said effectiveness. And most of the vaccines, actually, the least one is 50%, but most of them, they are above 70%, and some people claim 90% or 95%. But the effectiveness, what do they mean by effectiveness? They generate antibodies and memory T cells. That is the biomarkers, and I agree with them. Now, the safety issue is we don't know. If you take the first side effect, which is the alien side effect, these are nothing actually. Usually, if you have the reaction, either allergy or some sort of reaction, what we call in in the flu vaccination, because it's made in egg yolks, there is allergy to if somebody got allergy to the eggs, he should avoid them. While this is not the case in COVID, but the problem is how you measure the safety. A single death or serious illness, if proved, is too many. This is very important. And as you know, there are so far at least 8 skin reaction effects from the virus. So we know now there is an autoimmune reaction to the virus. Is it because of the live virus, or, which is quite rightly, it could be an autoimmune, which means that our cell will attack our immune cell, or immunity cell, attack our body cells. Next, please. How we measure effectiveness, as I said, in the clinical trial, at least 2 points. First endpoints is laboratory findings, T cell, anti-spike protein, and the other thing is the second endpoint. Are these trials actually prevented an outbreak? Because usually, I mean, now it took them to about 10 months, but usually it takes 10 years to make an effective virus, an effective vaccine, because one of their effectiveness, it shows that this year, for instance, we have less flu virus or less polio by testing them in the field. So far, we rely on biomarkers. Next, please. Next slide. So I think the pros and cons, and I talked about the cons, the pros, but the cons, the vaccine. What are the cons of the vaccine? The vaccine may lose its effectiveness. How long so far? Say 2 or 3 months. Some people say a year. We don't know. And there is a good reason to believe you can have antibodies against the vector, especially if you use adenovirus, human adenovirus, but some of the researcher or vaccine, they use chimpanzees adenoviruses, which we don't have immunity to them. Well, that will not affect the immune system. Now, as I said, this may produce short-lived memory cells as well. Now, there is a big issue, which I had mentioned already, which is antibody-dependent enhancement of the infection, like dengue fever. And it is important to recognize, just in case we don't know, if you have, going back to the question, is it safe or useful to have the vaccine if you have already experienced COVID-19? We don't know whether it's going to be boosting your immune system or you will suffer high because they call it augmented immune response, or ADE, which I already explained when I talked about the antibody opsonization. the virus has the ability to evade the immune system, which has been very well known. That is the reason why we have such a long incubation period, because the virus hides somewhere, probably inside the cells, and after repopulate, attack the immune system, and he got a cytokine storm. And to support that, they have high antibody or anti-S protein in those who are severely affected. Autoimmune antibody, I just mentioned, or the side effect of the vaccine may actually ameliorate the symptoms, so we will have a symptomatic carrier, which are very good in transmitting the disease. So these are all skeptical, arguments. Only time will tell. Next, please. So why the speed? Why the government decided to do that? As you know, because it's economic, political issue here, saving life. It's an emergency situation, and it's a lot of pressure on the government to act quickly, because infection control is a behavior science, and as you know, when the people do not follow infection control, you'll get many cases. The MRHA got a license. They call it flexibility license. They obtained it in April because of the grave situation that they can bypass the usual rules and accept or license the vaccine. This question, why it takes to 10 years to make a use of vaccine rather than 10 months, this is one of the, people always ask about this question, and we can tell them only time will tell. Next, please. Now, back to the original question. If I am cured from COVID-19, do I need vaccination? Is it safe? Or is it more protective, more antibody, more memory cells, or produce more harm due to ADA, autoimmunity? I've already, talked about it. Next, please. The answer, which actually the end is time will tell. I mean, this is all the answers about the clinical trial, and depends on the statistical design, the power of how robust is the study, and the first endpoint, which is biomarker, second endpoint, which is the clinical effectiveness. And now the other thing, which we found actually in other issues, infection control. So when we get a vaccine, because people now very much more educated about the risk, is it later, a year or two? Is it because we are good behaving, or because of the vaccine? And I can give you an example. When the joint replacement in the theaters, they said you have to have ultra clean air. And you can't do a joint replacement because that will you have a clean air. Then when came the issue of perioperative antibiotic prophylaxis, the study shows that, in fact, if you give perioperative antibiotics, you don't need ultra clean air. So the answer, time will tell. I think this is the final slide. Next. Thank you for listening to me.
Thank you so much, Dr. Khalid Alshafi, for an interesting lecture. Now,